Hi everyone, welcome to the Dry Lab stream. In this video, I'll give a quick overview of what's to be covered in the Dry Lab modules. For those of you that are new to Dry Lab, I'll just quickly say that Dry Lab in synthetic biology refers to the use of mathematical, physical, or computational models to study synthetic biological processes. The instructors for the Dry Lab stream are Braden, which is me, and Kavya and TNU. Each of us have different focuses within the Dry Lab stream. So I'll now briefly overview the three focuses of the Dry Lab stream. My content focuses on the fundamentals of modeling and systems in synthetic biology. And I've included this figure here of this person looking fairly distressed at their whiteboard of mathematics. And so the goal of these fundamentals lectures are that hopefully you'll feel significantly or at least slightly less distressed than this person appears when you're developing your mathematical models of biological processes. But jokes aside, the point of these fundamentals modules lectures are to give you just a basic toolbox of mathematical concepts that are useful in studying biological processes in general, as well to have a look at some biological processes that are really kind of ubiquitous and fundamental to get a grasp on modeling in order to do any modeling in biology. And the second focus is bioreactor modeling, which is going to be the focus of Kavya's content. This will look at some case studies and looking at specific applications that have occurred in real life of synthetic biology solutions at scale in bioreactors, as well as some foundations of actually modeling bioreactors, including mass energy balance. And for those of you who don't know, bioreactor essentially just refers to a device or system that facilitates a biologically active environment. And so this can be useful in using synthetic strains of, say, bacteria to produce an industrially or pharmaceutically perhaps relevant chemical compound. And this process in general may be more environmentally sustainable or efficient than traditional chemical approaches to producing these. So that's one application of synthetic biology, and we'll have a look at actually modeling these bioreactor processes in this module. And the third and final focus is TNU's focus, which is machine learning and algorithms in biology. And this will include looking at physics-based methods for molecular dynamics simulations, as well as looking at some machine learning algorithms, their applications, and their mathematical foundations. This will include maximum likelihood estimation, Bayesian optimization, variational autoencoders, and a few other things. Okay, so now I've overviewed the three different focuses of the Dry Lab stream. And now I just want to say, sort of as a disclaimer, that there are many potential applications of synthetic biology, and we're obviously not going to look at all of these applications. And so I've included these figures here, one of a field to represent agriculture and the other one of a bunch of pills to represent the pharmaceutical industry, which are two realms that synthetic biology can potentially have application in. And so I just wanted to sort of point this out as a way of saying that the Dry Lab stream modules shouldn't be considered a comprehensive overview of the modeling techniques that are required in the range of applications that synthetic biology can have, but more so just to give an example of some applications and ways to model different aspects of biology that are relevant to those unique applications, and to give you really a foundational kind of way of thinking and approaching modeling these problems and considering how to develop models in different realms. And similarly, there's an innumerable amount of biological phenomena that could be modeled. And so the application thing aside, just looking at fundamental biological processes that one could try their hand at modeling, it's just quite vast. And I've included all of these figures of some different aspects of biology, including protein and some DNA, and then we've got a colony of bacteria that are expressing some fluorescent proteins. and then a human brain representing an organ, and then we've got trees representing species. Like, so this is just to point out that biology spans a massive range of physical length scales, but also just is really diverse in terms of the phenomena and that could be studied, and each of these different aspects and facets of biology might be amenable to different computational and mathematical approaches to study. So again, the dry lab stream module shouldn't be considered as a complete overview of all of the things that one needs to know to model different biological processes, and certainly not an overview of all of the different types of biological processes that we could model. That's just far too ambitious. So I just want to say this as sort of to define the scope of these modules. And so in summary, you should consider the dry lab stream module as an introduction to mathematical modeling in biology or synthetic biology. And hopefully by the end, you'll have some basic ideas of the mathematical tools that are commonly used, some ideas about the foundational biological processes that show up almost everywhere when you're modeling biology, specifically those that are particularly relevant to synthetic biology and modeling cellular processes, and an idea of some applications and ways that these models translate to understanding the applications. And so now, just before I wrap up, I wanted to discuss some recommended readings from the Dry Lab instructor team. There's quite a few here, so I've color-coded them, as can be seen in the legend at the top, to indicate which focus that they correspond to. And so the blue ones correspond to my material, the modeling fundamentals, and the sort of orangey rust color corresponds to the machine learning and algorithm material from Tianyu, and the darker green color corresponds to the bioprocess and reactor modeling and metabolic engineering material from Cavia. So I'll just quickly go through some of the readings just to give you an idea of what they are. So the first one that I've included, Predictive Biology, Modeling, Understanding, and Harnessing Microbial Complexity, published in Nature Reviews Microbiology this year. I think it's a really great review for people coming from any sort of degree of knowledge in mathematical modeling of biology. It does a great job of talking about the development of mathematical modeling in biology and progress over the past sort of few years, but it also gives a really nice summary and definition and overview of different modeling concepts. And so I think that can be really beneficial to people that are fairly new to modeling even. And the second reference I've included is Mathematical Modeling and Systems Biology, an Introduction. And this is a textbook by Professor Brian Ingalls from Waterloo, who is one of our CS Berg advisors, actually. And this will be actually the main textbook that I use as sort of a reference for my modeling fundamentals material. So you might find that useful to follow along at some point. And the other textbook I've included, one of the other textbooks, is called An Introduction to Systems Biology, Design Principles of Biological Circuits by Yuri Allen. And this one can be considered as sort of a more advanced reference. And so you might want to dive into this if you want to go a little bit beyond the depth of the material that I cover in my modules. Although I do draw some things from this textbook in my content. And another textbook is Nonlinear Dynamics and Chaos with Applications to Physics, Biology, Chemistry, and Engineering by Stephen Strogatz. That's sort of a mathematics but textbook, but focused on applications more or less on dynamical systems and chaos. And so again, if you want to go a little bit deeper than the dynamical systems theory that we talk about in my lectures, then you might want to check out this textbook. And the last textbook that I've included here is by Pinsky and Carlin, called An Introduction to Stochastic Modeling. And again, if you want to go a little bit beyond what we've done in the stochastics module, the last module that I give in my content, then you might want to check out this textbook. But that said, it's still not a super advanced reference, and I think, in my opinion, it is still a fairly accessible textbook for people coming from different depths of mathematics backgrounds. And I've included this correspondence called Can a Biologist Fix a Radio? Or What I Learned While Studying Apoptosis. It's a correspondence to Cancer Cell Journal. Note its date. It's from 2002, so it's quite old, but it is interesting because it's around 2 years after when one might say the birth of synthetic biology was, around the year 2000. So just keep that in mind as it contextualizes it, but I think it makes it interesting. The author basically reflects and considers their experience as being an apoptosis researcher around this time, and it does this through an analogy with engineering a transistor radio, and basically kind of asks the question, is the nature of experimental biology and the way in which information is understood and learned in experimental biology amenable to gaining the understanding needed to actually engineer something? And so in this analogy, they look at how a biologist might look at the problem of fixing a radio. And in some ways, they argue that some work needs to be done to change the nature of experimental biological inquiry if we are to be able to engineer biology. And so I think this is interesting because it gives us something to think about as quantitative scientists in the realm of biology, and how we can actually contribute to that development of a new paradigm of biological inquiry that is amenable to gaining understanding of biology that is needed in order to actually engineer it, which really is kind of what synthetic biology endeavors to do. Now I'll move on to the machine learning references. So the first one is a link to a GitHub page that is for a third-year computer science course called Introduction to Machine Learning, offered at U of T. And I think there's some slides there that you can look at, and TNU has suggested that those might be a useful or interesting reference for folks. And then there's a textbook here that he's asked me to link you to called Machine Learning, a Probabilistic Perspective. And then there's a couple papers here that he said might be particularly relevant to some of the things that he'll be talking about in his content. And now moving on to the bioprocess and reactor modeling in metabolic engineering. There are two textbooks that Kavya has asked me to include here, one called Biochemical Engineering, and the other one called Analysis, Synthesis, and Design of Chemical Processes. And so you may want to check these out as a reference or guide or something to go a little deeper than what's covered in the bioprocess, bioreactor content. And then finally, there is a link here to a video from a metabolic engineering lab. This, I think, sort of like a Khan Academy sort of style video that'll introduce you to some concepts of metabolic engineering. And so you may find that useful as well. Just before I wrap up here, I want to say, please leave any questions, comments, or suggestions in the comments section below, and we'll get back to you as soon as we can. We really appreciate any of these, and it'll help us develop our material and improve it in the future. And finally, biology is neat and math is fun, so enjoy this, and I hope you learn something from it, and I hope that if you already don't love applied mathematics and modeling biology, maybe you'll like it a little bit more after this. Thanks for listening.